Hello friends, today we are going to see what are polysaccharides and different examples of polysaccharides. So let's first see what is polysaccharide. Polysaccharides are formed by linking large number of monosaccharide units by glycosidic linkage. It is a kind of carbohydrate. When many monosaccharides combine, they will form a polysaccharide and they are linked by a glycosidic linkage. Then second point, examples starch, cellulose, glycogen are the most common natural polysaccharides. Starch is storage carbohydrate of plants and important nutrients for humans and other animals. Cellulose is the main constituent of cell wall of plant and bacterial cells. It is also main constituent of wood and cotton. Glycogen constitutes storage carbohydrate of animals and is present in liver, muscles and brain. It is also found in yeast and fungi. Now let's see starch in detail. So starch is a polymer of alpha D glucose. We know there are two forms of glucose, alpha D and beta D glucose. So starch is a polymer of alpha D glucose. Starch has two components, amylose and amylopectin. Amylose is around 15 to 20%, amylopectin is 80 to 85%. Amylose is soluble in water and forms a blue colored complex with iodine. It contains 200 to 1000 alpha glucose units linked by alpha 1,4 glycosidic linkages, giving rise to unbranched chain of variable length. Amylopectin is water insoluble component of starch which forms blue-violet colored complex with iodine. It is branch chain polysaccharide. In amylopectin, chains are formed by alpha 1,4 glycosidic linkages between alpha glucose, whereas branches are formed by alpha 1,6 glycosidic linkage. So that's the difference between amylose and amylopectin. Amylopectin is having alpha 1,4 as well as alpha 1,6 glycosidic linkage, whereas amylose is having only alpha 1,4 glycosidic linkages. Next up is cellulose. Cellulose is a straight chain polysaccharide of beta glucose units. In starch we saw it is made up of alpha glucose units. Here in cellulose it is made from beta glucose units linked by beta 1,4 glycosidic bonds. Chemical hydrolysis of cellulose requires use of concentrated strong acids at high temperature and pressure. This implies that the beta 1,4 glycosidic bond is very strong and difficult to hydrolyze. That's why we cannot eat cellulose. So one question is regarding that only. Cellulose cannot be digested by human beings. Justify. In cellulose, beta 1,4 glycosidic linkage is present and it is very strong and difficult to hydrolyze. Humans do not have enzyme which can hydrolyze this linkage. Therefore, cellulose cannot be digested by human beings. Now next up is glycogen. Last one, its formula is C6H10O5 whole bracket N. It is stored carbohydrate present in animal body. It is also known as animal starch because its structure is similar to amylopectin, but it is rather more highly branched, or we can say it is more complex alpha D glucose units. It is present in liver, muscles and brains. It is also found in yeast and fungi. Glycogen breaks by the action of enzymes to glucose on demand of the body. So that's all from polysaccharide. So we saw different examples of polysaccharide today, including starch, cellulose and glycogen. Thanks for watching the video.